Hi, welcome back to the CD Genomics Next Generation Sequencing video series. In this video, you will learn the overview of microsatellite and microsatellite genotyping. If you want to gain a deep understanding of microsatellite genotyping, please visit our website. This video falls into 4 parts. What are microsatellites? Why microsatellites are valuable markers? Microsatellite genotyping, and applications of microsatellite genotyping. Microsatellites, also known as short tandem repeats, simple sequence repeats, or simple sequence length polymorphisms, are found in both prokaryotes and eukaryotes. They are tandem repetitive sequences, in which the repeating unit contains from 1 to 10 nucleotides. The number of repeats is variable in populations, since microsatellites are prone to slippage during DNA replication. Despite the microsatellite instability occurring in all dividing cells, microsatellites remain stable in length. Why microsatellites are valuable markers? First, they are widely distributed, across the genome, including encoding and non-coding nuclear and organellar DNA. Second, they are highly polymorphic. The reason seems to be slippage replication. Third, they are locus-specific. Locus-specific primers allow us to amplify locus-specific microsatellites. Fourth, they are codominant. Heterozygotes can thus be distinguished from homozygotes, in contrast to amplified fragment-length polymorphisms. Fifth, PCR-based methods can be used to analyze SNPs, which do not require expensive laboratory instruments. Sixth, SNP analysis can be used for a range of applications, such as individual ID, and phylogenies. Microsatellite marker polymorphisms have replaced amplified fragment-length polymorphisms, as the markers for constructing linkage maps. Genotyping is the process to identify the genotype of each microsatellite. The workflow of microsatellite genotyping generally involves specific primer design, amplification of microsatellites, and polymorphism testing. If someone has developed SSR primers for a closely related species, these primers will be well worth checking in your species. If, however, there are no primers developed for related species, you may need to design your own. It is necessary to have the nucleotide sequences first, either from your own sequencing project, or from the NCBI nucleotide database. SSR motifs can be identified, using the microsatellite identification tool. Locus-specific SSR primers can be designed with SSR locator. Once the SSR primers have been developed, it is necessary to validate these primers using PCR amplification. Each SSR-PCR reaction mixture consists of DNA polymerase, reaction buffer, magnesium ion, DNTP mix, forward primer, reverse primer, and genomic DNA. Thermal cycling is performed in a PCR instrument, under the following conditions: initial denaturation at 94 degrees Celsius for 5 minutes, 36 cycles of denaturation at 94 degrees Celsius for 50 seconds, annealing at optimal temperature for 1 minute, and extension at 72 degrees Celsius for 1 minute, as well as a final extension at 72 degrees Celsius for 7 minutes. The PCR results can be visualized by agarose gel electrophoresis, or polyacrylamide gel electrophoresis. Page genotyping is labor-intensive, but provides good resolution. For fine-scale studies, marked SSR primers can be synthesized with a fluorescence dye, for SNP genotyping by capillary electrophoresis. In this case, each PCR product is loaded into a capillary, containing a polyacrylamide matrix, in which the electrophoresis is performed. The emitted fluorescence is captured, and the molecular mass of the PCR product is determined. An electropherogram is then generated, which shows luminescence peaks, corresponding to each amplified allele. Microsatellite markers are very useful in the studies of biological diversity measured on the basis of genetic distance, to estimate gene flow and crossing over rates, to infer intraspecific genetic relationships, to construct linkage maps and to find cultivar DNA fingerprints, to map loci involved in quantitative traits, to determine the degree of kinship, in breeding using marker-assisted selection, and in disease diagnosis. That is, these markers are very useful in human health monitoring, agricultural production, conservation biology, and evolutionary biology. In the end, if you want detailed information about our microsatellite genotyping service, please visit our website, www.cd-genomics.com. We are more than happy to be of assistance.